The second speaker in our session is Alessandra Curioni. And Alessandra is the head of the Medical Thoracic Oncology Unit at the University Hospital Zurich. She's a research group leader in translational oncology and chair of the expert group in immuno-oncology. The major focus is on the development of therapies for thoracic malignancies through immunomodulation. And she was the PI of the first concluded trial with local delivery of CAR-T for mesothelioma. She's the PI of several trials that are focusing on immunotherapies and also has various leadership roles outside the UZH, including being coordinator of the lung cancer program of the European School of Oncology and the deputy head of immuno-oncology of the Swiss Group for Clinical Cancer Research. So I am very pleased to welcome Alessandra to bring in some deep clinical insight on this morning's topic. Alessandra.
Thank you very much, Shana, for the very nice introduction. And I will try to guide you through the challenges we are facing in the clinic when there is so much of innovation and at this pace of research. So I will share my presentation now with you. So it's very important to say, yes, I am a clinician, and yes, I lead a research team, but I have to say that to combine the two of them and at the pace what I was just mentioning before and listening up the previous talk into the clinic is not as fast as you do in research. And especially there are several challenges that I would like to share with you. This is my disclosure slide. So in order to share this experience with you, I was thinking to discuss about one particular type of cancer, and this is lung cancer. And why that is not only because it's my subspecialty, but especially if you look at the mortality rate in the darker bulk for men and
Alessandra,
women.
Sorry for interrupting. Can you swap the display?
Lung cancer is not only a very frequent kind of cancer, but highly mortal. And this is what you can see here with a darker bulk. And we will focus on one specific stage of disease, which is stage 4, when the lung cancer has metastatized to different sites. If we go back to some years ago, despite the use of different kind of chemotherapies, about 50% of patients with lung cancer in stage 4 would have died in 8 months. So how and was improved now the treatment of this disease? Let's go back to one of the first slides that was also discussed, one of the important steps of the cancer cells recognition by the immune system. We know that, and this will be important for the next step also for the discussion on treatment development. So we know that cancer cell can present on the surface antigen, and some antigens are presented by the HLA system. And by this, the T cell can recognize the cancer and destroy it. But there are ways by which the immune system can be suppressed. And an example of this system is through immune checkpoints. And this is one of the many immune checkpoints that are known, but that are fundamental for understanding the way cancer treatment has changed and revolutionized in the last year. And this is the pathway of PD-L1 and PD-1. PD-1 expressed on T cell, interacting with PD-L1, for example, on the cancer cells, will lead to a suppression of the T cells. Now, the use of immune checkpoint inhibitors has the possibility of restore this recognition by blocking these immune checkpoints. And this is what we're going to discuss now through. How do we do it in the clinic when something like this is recognized, and to understand if really such a treatment would work? We do it into clinical trials, and these are examples, the very important milestone trials for the development of immune checkpoint inhibitors in lung cancer. These are 4 different studies taken into account, including patients with lung cancer, stage 4, who did not respond anymore to a so-called first-line treatment. So now we have patients who did not respond to a first line and might be included in this trial by receiving either a PD-1 inhibitor or the conventional chemo. And these are other ones, so PD-1 and PD-L1 inhibitors. All of them show that there is a clear improvement in survival of these patients compared to the standard chemotherapy. And this led to the approval of many of those. So I'm not going to talk about all of them, obviously, but this is important to understand the pace by which the many different companies have developed drugs to target this pathway through the success of these treatments. And this is just an example that I would like to share with you. This is a patient who was treated in our unit who had lung cancer. You can see there is a liver metastasis, several lymph node metastases, bone metastases. Normally the bladder and the brain would also look black as well as the kidney. But what is important is that we gave chemotherapy to the patient and the cancer is increasing in size. So we switched the patient to treatment into immunotherapy, an anti-PD-1 antibody. And what you can see is a clear response of the whole tumor. What you see here is simply the heart, which is active, but the tumor nearly, we could say, disappeared in the images. And this is through the time. Why is that important? And you can see this in this graphic, is that patients who responded to immunotherapy, there is a percentage of them in this trial and in this trial here, they respond for a long time, which means that the immune system has learned, has a memory to recognize the tumor then over time. Please keep in mind that in these studies where patients who received maybe several lines of chemo, and here about 20% of them are surviving at 5 years, which is something we have never seen for lung cancer before. Now, who are the responders? Let me go back once. So who are these patients here? How we can recognize them? And maybe by doing this, we would understand also how to improve the treatment for the non-responders. So we have said before, there is this pathway that is known, and most of the clinical trials have focused on the presence of this molecule PD-L1 at the tumor site. Now, the difficulty we are facing in the clinic is that different drugs in different studies have used different antibodies to stain, for example, PD-L1. So, and especially the cutoffs are also different. And what we are looking at, either tumor cells or immune cell, might also be different. So this is a major challenge in the clinics that we are facing, especially if we want to translate the result of a study into the clinics. Clear example. So in these studies, patients with stage 4 lung cancer were included. They had tumor cells more than 50% positive for PD-L1 and received either immuno or chemotherapy. What you can see here is that the survival of this patient was much longer by giving immunotherapy compared to chemo. So probably using PD-L1 would be enough. Well, a difficulty that I would like to share also with the previous speaker is when we are looking at the tumor, and this is just a cut of a tumor, we might look at different sites. And you can see it here is a clear example of this tumor where in some areas the tumor cells are completely negative for PD-L1 and some other tumor areas are highly positive for PD-L1. So the clinician is faced, as well as I would like to share this challenge together with a colleague from pathology, where should we look at? So what we usually do in the clinic is that the colleagues of pathology look at different places, so different sites of the tumor, in order to make an average. But this clearly makes an example of how heterogeneous as well a tumor can be. And moreover, sometimes we look at tumor specimens that are from patients previously collected, for example, a biopsy done 1 year before or 2 years before. And here we also could see in different patients that an initial tumor might be completely negative, but when it recurs, it can be highly positive. So the positivity is by looking at the brown surface of the tumor cells. We can see it as well here. So a tumor which is highly infiltrated by immune cells, but very, very faintly negative, becoming positive at recurrence. So this tells us that we need to look at the tumor at the right time point and understand as well the heterogeneity. And not only PD-L1 is important, but it was mentioned before, looking at the antigenicity, so the presence of antigens on the surface of tumor cells can be very important. How can we look at it? So we know if we look at many different tumor types, that in tumors mutations are occurring. And measuring the amount of the total number of coding somatic mutations in a tumor genome is called the tumor mutational burden. Why is this important? Because the more the mutations are occurring, the more the neoantigens might also occur in that tumor. It has been correlated for different tumor types. Each dot here represents a different tumor type, that the highest the amount of somatic mutations, the highest is the response to immunotherapy. And clear examples are cutaneous squamous cell carcinoma, melanoma, and subtypes of colorectal cancer. But we need to understand how this likelihood to respond to immunotherapy occurs. One issue is that, true, the highest is the immunotherapy benefit to immunotherapy, the highest the ratio of mutations. But how do we define such cutoff of so-called high, intermediate, or low? I have to tell you as a clinician, this is the next challenge we're facing in diagnostics. And this is due to the fact that many different groups in different places are using different ways of looking at this mutational burden. There is the Cancerplex, the Oncomine, the MSK, and so on. The foundation one is what we are commonly as well using in the clinics. But they are looking at different amount of genomic content, and they're not always overlapping in the terms of mutations, which means we will have a value of how many mutations are there. We might not have a quality of which mutations, and this might be important to have a consensus. This could translate in, for example, looking at a population of patients and the amount of response to immunotherapy, and combining the presence of tumor mutational burden and PD-L1 expression. Taking this information together probably might tell us who are these patients. But still it's not enough. We're not reaching the 100%. Why that? Is because, of course, a solid tumor is highly complex. There are not only the tumor antigens, there is an immune suppressive environment which might and should be analyzed. There are the inflamed tumor, but there is also the host environment that we should take into account by doing this broad spectrum analysis. And it has been a very interesting talk, the one given at the beginning from Professor Bassani, to understand the complexity of all this information. And these information are not important only when we take the decision of the treatment at the very beginning, but also in, I would like to say, the three major challenges that we're going through as clinicians. The one of the resistance, the presence of pseudoprogression, and I would like to share some information about this with you, and also the side effects induced by immunotherapy. What is resistance to immunotherapy? We might have a resistance by ab initio, a tumor not responding to anti-PD-1 treatment, just grows. Or, for example, the so-called acquired resistance. Patients, many tumor sites, responding to immunotherapy, but then at what site? This site is resistant. So this is very important, because if we would understand why this is occurring, we would also be able probably to implement the treatment at the very first beginning. There are several ways that resistance have been looked at, the primary adaptive or the acquired, by looking at escape mutations in the interferon gamma signaling, loss of antigen expression, loss of HLA. For example, we are looking at it specifically, and Dr. Hilbrunner in my lab is focused on looking exactly at the biopsies of tumor, at the samples of tumor of patients when they responded to treatment and at the second time point, and we're looking at different insights to that in lung cancer. We could also maybe do a combination treatment without a specific rationale, but by using a broad chemotherapy, we might reach, as it has been seen for many different trials, that giving the addition of chemotherapy might improve and might synergize with immunotherapy as well. This can be also performed by the combination of several co-stimulatory signals or by inhibitory signals or by targeting the network. And of course, I'm not going into details, but I'm mentioning this because in the clinics, there are several clinical trials ongoing exploring, and you can see it here, more than 2,000 are described in this review, but there are many more going on, describing the combination of immune checkpoint blockade with anti-PD-1 or PD-L1 without either immunotherapy, chemotherapy, and many different and other attempts. There is also the issue, and I would like to look at it carefully because it was not very well characterized, why patients, and each one of these lines represent a patient, might have an increase of size of tumor and then a decrease of size of tumor. We're looking at the size, not at what is occurring at the tumor site exactly. And this is probably because when we have a tumor and we give immunotherapy, such tumor becomes bigger because of the reclaim of the immune system and then diminish over time. But in the clinic is a major challenge. This is the case of a patient with multiple metastases from lung cancer that after immunotherapy had many new spots occurring. And this was described to be a possible progression. So what we did at the time point is that we biopsied all of these lymph nodes to understand if we had to change treatment or not. And we could find a clear inflammation at the site with several CD3 and CD19 positive T and B cells. And after 3 months, the patient had a complete remission and is still in complete remission. We still need to understand this challenge. And this is very hard when we're looking, for example, at the brain. Would you say that this lesion here is a metastasis and this huge lesion here in the brain is not a metastasis? It is exactly like this. This is because by using a specific target, and I've seen that there will be also colleagues from Professor Shibley giving a talk tomorrow about tracer for PET imaging, we could really characterize the tumors or the immune system that is coming at the site. What you see here is the immune system coming at the site of a metastasis to kill the tumor. And after a while, it disappears. Still, another matter of interest for the clinic is that patients do develop side effects to immunotherapy. You might imagine as this, what I'm used to say to patients, we switch on an immune system and with not specifically, so they might develop, in this is reported in many different clinical trials, side effects of different kind and different grade. And these can occur at many different sites, from the bone to the joints, from the adrenal glands, in the gland themselves, thyroid glands, and the liver, and so on. And this is something that we are facing every day, and we would like to understand why some patients are developing these side effects and others not. And many research are ongoing. There is a very interesting collaboration together, for example, with the cardiology department here at USZ, led by Professor Matter, to look exactly and understand why myocarditis are occurring. And to conclude, I would like to share with you just one case to understand the importance of this knowledge. We treated a patient who was under immunotherapy, and she was very tired and started to lose weight. So we thought the cancer is spreading, was not the case. She had an infection, was not the case. Not even an issue of the thyroid or the hypophysis. So it was a very simple lab test that helped us to understand what was going on. And this is by testing the sugar. And I have to say that we missed the first alteration of the sugar because the patient was not fasting as well at that time point. And we checked it again when the side effects occurred, and we could see this extremely rapidly increase of sugar. Why that? If we would have gone back to the first publication and how the PD-L1 pathway have been discovered, and the Nobel Prize was given, as you very well know, to the group who discovered this, we would have understood immediately what was going on in the patient. And this is what we need to understand when we see side effects. We need to go back, try to understand what's the mechanism there, and probably we might prevent or we might understand which patient will develop such side effects. And in this case, it's because pancreatic beta cells to express PD-L1, and when we gave the blockade of the treatment, we have led to a diabetes. So by this, I would like to thank you all very much for your kind attention, and I'm available for any questions. Thank you very much.